for use. There are many parameters like pH, color, COD, BOD and are tested in the laboratory. So the taste for total coliform and fecal coliform, there are two methods to detect total coliform and fecal coliform in a sample. One is AFA method and another one is IS method. So the coliforms are water contaminant organisms which are mainly from the bacterial family called Enterobacteriaceae. This stage detects coliform bacteria which indicate possible contamination of water. The AFA 9221 multiple tube fermentation technique is used for estimating total and fecal coliforms. The reagents which are used are LT broth, BGLB broth, McConkey agar, EC broth. The equipments include laminar airflow cabinet, autoclave, micropipettes, Durham's tube, and incubators. There are various other equipment required which are listed below. The procedure for the test. So for that, firstly we have to collect the sample. So the samples are collected in sterile container and refrigerated at 4 degree. Water samples are inoculated firstly into the LT broth tubes and incubated. Gas production in the Durham's tubes indicate presumptive presence of coliform bacteria. The coliform bacteria produces gas and acid in the Durham's tube. The gas is which is produced by the coliform bacteria is mainly CO2 gas. Conform taste. Positive tubes are transferred to BGLB broth and EC broth for further tasting. The gas production confirms the presence of total coliforms and fecal coliforms. Furthermore, the MPN values are calculated. Completed taste. For the completed taste, the cultures are streaked on McConkey agar plate. Typical lactose fermenting colonies are further examined by gram staining. Gram negative non-spore forming rods confirm coliform bacteria presence. This is a picture of McConkey presence of gas production and presence of gram-negative non-spore forming rod-shaped bacteria shows the presence of coliform group. So now Ashvi will carry the presentation forward.
IS method is an another standard method for detecting total and fecal coliform. The principle remain the same, but McConkey broth is used as the primary medium. The procedure include presumptive, confirm, and complete phases. These in this method, we use equipment like laminar airflow, autoclave, micropipette with autoclavable tips, refrigerator, test tubes, petri dishes, slides, etc. The procedure remains same, but the media we use McConkey broth in this IS method. Confirmed phase in this the from all the positive tube from the presumptive test, take a loop full of culture and inoculated to in brilliant green lactose bile broth and incubate for 37 degree for 48 hours. Simultaneously, inoculate loop full culture in EC broth and incubate at 44 degree for 24 hours. In complete test, use McConkey agar plate and BGB broth showing gas and incubated 37 for 24 hours. You can show in the picture the gas production in McConkey broth and lactose fermenting colony on McConkey agar plate. In the interpretation, presence of gas production and presence of gram-negative non-spore forming rod-shaped bacteria show the presence of coliform group. This flowchart explain the IGES method. We perform another parameters known as DO or BOD, dissolve oxygen and biochemical oxygen demand. The aim of this experiment is to assess water quality by measuring dissolved oxygen and biochemical oxygen demand, which indicate the level of organic pollution in water. Requirements are the experiment requires dilution water, incubation bottle, nutrient solution, diometer, and an incubator for maintaining suitable condition. Introduction. BOD represents the amount of oxygen consumed by microorganism while decomposing organic matter. The test measures oxygen depletion under controlled aerobic condition. In the procedure, dilution water is prepared by adding buffer and nutrient solution. Samples are diluted appropriately. Initial DO is measured by diometer. Bottle are incubated for specific period before measuring final DO. Incubation. Sample are incubated for 3 days at 27 degree for 5 days at after incubation final DO is measured and the difference between initial and final DO is used in BOD. BOD is calculated using different uh, between initial and final dissolved oxygen value with correction seed control when required. Thank you for your attention. This internship provides us valuable hands-on experience in environmental microbiology, water quality analysis, and laboratory technique used for the pollution monitoring. Thank you.
Okay, so very good presentation. Um, what is the meaning of AFA method? Abbreviation. What is the full form of APHA?
Ma'am, AFA is the American Public Health Association.
Okay. And what is fecal coliform?
Fecal coliforms are the bacteria which are found in feces of animals, warm-blooded animals.
Okay. Okay. And what is LT growth?
It is luryl tryptone broth, which is rich in tryptone amino acid, which is an aromatic amino acid.
Okay. So, have you performed any analysis in central laboratory?
Yeah, ma'am, we have done that experiment, and the pictures mm, we attached in the presentation were done by us.
Mhm. And when will you submit your final report?
Ma'am, this Monday we will submit it.
And what you have learned from your this analysis experiment?
We have learned about that how the bacteria contaminates our water, and it is very harmful to drink the contaminated water.
Mhm.
And it is very important to manage the water contamination.
Mhm. Do you know about MPN? What is MPN?
MPN is most probable number.
Yeah, what is that? It is the full form, but what is that?
It is a statistical estimation of concentration of coliform bacteria in a water sample, which is determined by the gas production in the Derham's tubes.
Okay, okay, fine. Dhruvi, from your side.
What is the standard limit or a permissible limit for the TC and FC in the drinking water and the discharge water?
It is. It should not be more than 1,400.
For both, it is same.
No, for drinking water, I guess. I have know about. I don't have idea about that particular number, but it is around that.
Okay, but make sure you learn about this.
Okay, ma'am.
Okay, why we can't use coliform test to detect virus and protozoa?
Viruses cannot be cultivated on these mediums.
That's the only explanation.
That's the much I knowledge have, ma'am.
Okay.
Who is answering all the questions? Sorry to interrupt. Who is answering all the question? Only Kavya only?
Yes, ma'am.
What about Ashvi? Let give her chance to answer the question.
Oh, okay, ma'am.
Ashvi, if the total
Yes,
coliform
ma'am.
is high
Hmm.
but fecal coliform is low in the treated effluent, so what does this treatment process suggest to us?
Can you repeat the question, ma'am?
If the total coliform is high
Hmm.
but fecal coliform is low in the
Hmm.
treated effluent, so what does this treatment process suggest to us?
If an effluent sample show high total coliform but low fecal coliform, it indicates general bacterial contamination in the sample.
Can you repeat the answer?
Yes, ma'am. If an effluent sample show high total coliform but low fecal coliform, it indicates general bacterial contamination, but the contamination may not be primarily from fecal source.
Do you know any treatment processes for this?
Chlorination, UV disinfection, ma'am. Membrane filtration we can use.
Do you know any health hazards if this kind of bacterial microbial present in the wastewater? So what kind of health hazards to the human being?
Gut related issue and skin infection.
Do you know Ashmi, any permissible limit for TC and FC?
No, ma'am, I don't have idea about that.
Okay.
Okay, done.
Yes.
Dhruvi?
Yes, ma'am.
Okay. Okay, fine. So, any other presentation? બીજા કોઈ ઇન્ટર્ન્સ કરવાના છે પ્રેઝન્ટેશન?
ના આ બે મારા ધ્યાનમાં હતા કાવ્ય અને અશ્વી બાકી અત્યારે કોઈની પતિ નથી રહી.
Okay. So, let us conclude this session. Okay. તમે સ્ક્રીન શેરિંગ સ્ટોપ કરી શકો છો કાવ્યા. So we are ending this induction training here. So dear student, I hope that this training has helped you understand the importance of your internship and the practical role of environmental professional in pollution control and environmental protection. And further, you are requested to sincerely follow all the internship instructions, submit your reports and presentation on time, maintain discipline, and make the best use of this learning opportunity. And another important instruction is that use of chat GPT is not bad thing until you know the meaning of all the things whichever you have mentioned in your report and PPT. But if I find that you have used ChatGPT and included all things in reports, but you have not knowledge about that, so I will terminate your internship. Okay. So mind that whatever you have written in your report and PPT, you know the meaning of it. So thank you everyone for your active participation. And with this, we formally conclude the online induction training program. Anyone have any question and doubt? So we have 5 minutes. You can ask me. Okay, so let us end this induction training here. Wish you all the best and make the maximum utilization of this internship opportunity. Have a nice day.